Okay, if everyone could take their seats, it's 1:15. I hope you all had a very nice lunch and had some time outside if you weren't listening to the excellent presentations that were going on here during the lunch break. Our next session, we have one speaker, and we're very lucky to have that one speaker, Kirpalani, who spoke yesterday, and I won't go through his past history again, but I think it's the right person to help address a question which for many of us in critical care, I think needs to be continually readdressed, and that is designing clinical trials in critical care. Do we have to have a rethink? I think that's a fantastic topic to spend some time pontificating upon maybe, or at least considering, because our track record is not as good as often we think it is. So I'm really looking forward to hearing what Haresh has to tell us, and he's got about 40 minutes to solve all of the problems for neonatal and paediatric ICU trials. So thank you very much.
Good afternoon, everyone. First of all, many thanks again to Peter and his coordinating committee. It's a great honour to be here. Secondly, I must express my regret that you're having to listen to me instead of Alan Jobe, who was scheduled to give this, but Alan was unfortunately unable to do this. And I would have loved to hear what Alan made of this topic. This is the topic that Peter has asked me to talk on, and indeed it's a little bit challenging. But I think in our group of communities, and I'm cognizant that there are several communities here, we do need to think about these issues. So over the next 30 minutes odd, I'm going to try and address three topics. I'm going to remind us, first of all, why on earth we need clinical trials. And then I'm going to consider briefly how we've done. We've come along the road from authoritarian anecdotal medicine towards something that calls itself evidence-based medicine. Well, how have we done? And in the space of time that I have, I'm only going to focus on two items: the size and quality of the current trials. And that should set us up for what Peter and his committee particularly emphasized that I try and address. And the pivot of this is going to be Bayesian analysis. I offer a disclaimer right now. It's my only disclaimer. I am not a statistician, but I think even I can grasp some of that, so that means you can grasp it. And that's the pivot for things like adaptive trials and platform trials, and we'll try and go there. I mean, I think that certainly the older members of the audience will recognize the air of authoritarian medicine that comes across in this slide. This is a painting, and it's supposed to be, it's not certain, but it's supposed to be Professor Charcot. And he's doing an ear auscultation, an examination of this patient, and you can see that he's surrounded by a rapt, attentive, predominantly, overwhelmingly male audience. And I would venture to say that no one asked him what is the diagnostic utility of ear auscultation. As we moved into the 50s, we began to be a little bit more questioning and began to ask questions. And David Sackett, in adult medicine, one of the acknowledged founders of evidence-based medicine, actually said that Bill Silverman was really the prime exponent of evidence-based medicine. And Bill was a neonatologist at Columbia. And in this book, which if you haven't read, and I suspect some of the younger members of this audience have not read this, it's available free online at that website. And he looked at the proclaimed therapies that there were in neonatology at the time. And he divided them into some subcategories. You can't see them here, so I've expanded them here. These were really based on physiology, and he devised three categories: either sound of practice, or it had led to disaster, or it had led into fruitless byways. And I've highlighted the therapy of oxygen for periodic breathing. And you can see in the respirogram here that the advent of oxygen to this baby certainly ablated the periodic breathing related to apnea of prematurity. But it came at a cost. It came at the cost of blindness and retrolental fibroplasia. And that's why he categorized it as a disaster. And that's why he led some of the first pioneer randomized trials of oxygen in the baby population. Now, you would think that our peers would have adopted the mentality that this made a lot of sense, but you may have forgotten how difficult a struggle it was to get evidence-based medicine on any platform. And as recently as the early 2000s, there were satires. They posed as satires, but they were really mocking the mentality of evidence-based medicine. And the argument was that no randomized controlled trials of parachute use have been undertaken. And therefore it was silly to expect randomized trials for every therapy. The problem is that these authors had radically simplified matters. The first thing is that no methodologist worth his or her salt would advocate such a thing, because there's no doubt about what would happen. It's only when there's doubt about a therapy that you need to subject it to randomized trial. Moreover, in medicine, there are very few things that have 100% risk of death or a bad outcome. And there's a therapy that is almost 100% or 100% valuable in averting that outcome. So since 2003, many more people, as you know, have adopted randomized trials. But there's another variant that pops up, and we're all in the intensive care of various settings, largely because we love physiology, I suspect. I think it's that way round. It's not exactly a causal relationship, but I think that people who like physiology are attracted to intensive care. And a variant of the skepticism around evidence-based medicine comes from the physiological argumentation. Now, I want to be very careful that you don't misunderstand me. I'm not saying physiology is not important. In fact, it's fundamental to framing a question, but I'm going to argue that it's not sufficient to prove a therapy that still needs solid trials. And what I've tried to do here is in the second column depict animal physiology findings for various studies that have been also tested in human trials. So the first line is antenatal steroids to prevent RDS. So a thumbs up means it's good, obviously, and it's borne out in human trials. The bottom line is surfactant, and also thumbs up between animal physiology findings and human trials. What about other things? This is not comprehensive. These were just the ones that quickly came to mind. Caffeine. In asphyxiated animals, it's a bad thing. It enhances anoxic injury in the brain, but when given to infants with apnea of prematurity, it's a good thing because cerebral palsy is reduced. And similar findings can be adduced for sustained inflation and hypothermia. Hypothermia, there's both a down and an up, and people get confused about that, but they've forgotten that Jeffrey Dawes did a lot of animal experiments in round about the 60s, I think, with macaque monkeys and put hypothermia for perinatal brain injury on hold, finding a worsening of effects. So again, the message of this slide is essentially that physiology is very important, is fundamental to designing a solid trial, but it's not sufficient of itself. I think we just have to think about all of the arguments in neonatology, even today, about whether or not to close all patent ductus arteriosus in preterm infants. It just rages on and on. So it's not quite dead, this anti-trial mentality. But by and large, we have moved away from the era of authoritarian medicine. And we are in an era of evidence-based medicine. How has that gone? How have we done? I'm going to focus only on two items. Firstly, the size and the quality of current trials. I mean, it goes without saying almost that size matters, as these crash dummies are about to learn very shortly. The number of randomized trials in neonatology has rapidly increased. I'm not going to show you that data, but what I am going to show you is a data slide from Suzanne Hay's work, which is basically showing you the median and the interquartile ranges of sample size in randomized trials from 2003 up to 2016. It hasn't changed much after that. She and John Sapanchik are in the process of getting this data published. But it essentially tells us that the sample size is small. What about the quality of trials? A few years ago, 2011, Sarah DeMorrow looked at the quality of neonatal trials. And in her systematic review, she looked at trials that were published, first of all, on the left-hand side in pediatric journals, and those published on the right-hand side in the top medical journals, that is to say, New England Journal, JAMA, and Lancet. And on the x-axis is depicted the number of quality criteria that are derived from the grid of the Cochrane risk of bias tool. And as you can see, for the pediatric trials, about 60% are relatively good or perfect on that risk of bias score. But there's this long tail going downwards with unacceptable quality of trials, in my view. In contrast, those papers that reached the top medical journals, they all uniformly had either an acceptable or good quality ranking. My question to you, some of you are editors of journals. Why are we accepting substandard articles on randomized trials? In any case, the broader question for us is what to do about this. Now, the same situation, in case you're in any doubt, it's not just the Mickey Mouse neonatologists who are doing relatively small trials and perhaps poor trials. In adult ICUs, the trial situation is actually, to my eye, not that much better. This is from a systematic review in Journal of Clinical Epidemiology. And out of these almost 600 trials between 1975 and 2015, the top graph shows you the annual median size of those trials. And you can see there's a broad 95% confidence interval, but the median is barely over 100 as recently as 2015. This was updated to about 2018 in the final publication. And the second graph below is the percent of trials with a low risk of bias. And you can see that actually it's rather few, varying between, in the later years, between 10 to 25% of the adult ICU trials. There's similar data for the pediatrics, but I just wanted to anchor the general issue at the age ranges, at the extremes. Now, it's certainly true that we can still do, when the community wants and is able to do, a very high quality trial in the older style manner that's large and can give us meaningful information. This is the Paris 2 trial, which randomized children less than 4 years of age with hypoxemic respiratory failure to the receipt of either standard oxygen therapy or to the use of high-flow cannulae. And they showed statistically significantly and convincingly that the infants who were randomized to the high-flow nasal cannula had a longer stay in hospital. So we can do such trials. They're expensive. Perhaps we might want to talk about this. And perhaps we can yet still do better. So that brings us to really the heart of what Peter wanted me to try and bring out. I think the prologue was necessary because I think we did need to understand how we got to our current status and what are the limitations that we've got to. But now, having acknowledged that, yes, we've made some strides, where do we go from here? We've acknowledged some problems. How can we overcome them? More and more people are looking at adaptive trials and platform trials. But as I've said, the key to both of them is really a Bayesian approach. What do I mean by that? And what does the literature mean by that? I'm going to try and display this rather than a semi-didactic way of doing it, is to explore a critical trial that has had resonance. That is the so-called THACPA trial, a recent trial. THACPA stands for therapeutic hypothermia after out-of-hospital cardiac arrest. So I'm not talking about the in-hospital study that the same authors basically conducted. This is out-of-hospital. The PCOT of this trial was in comatose and ventilated children after out-of-hospital cardiac arrest between 2 days and 18 years, so a broad range. Does the intervention, the randomized intervention of therapeutic hypothermia, as opposed to standard care of normothermia, does that increase the survival? So it's a composite outcome, a survival with a good functional outcome. That's a modified Bracken score, as I understand it. Jeannie, if she's in the audience, may probably correct me, but I think that's what it is, and measured at a time of 12 months. They calculated a sample size of about 270 patients for an 85% power. And what did they find? These are the results. They recruited 295 patients, and they had 260 patients with evaluable data. This is something that's not surprising. We see that perhaps parents refused or moved away, were untraceable, whatever. Nonetheless, there was no significant difference in the primary outcome between the hypothermia group and the normothermia group at 20% versus 12%. So an 8% absolute risk difference, or benefit, if you will. Now, there was a broad confidence interval, 0.86 to 2.76, and accordingly, that led to a p-value of 0.14, which is obviously greater than what we consider a statistical boundary by convention of significance. So this is a quandary, right? I mean, if I were the parent of a child in this age group, in this situation, I think I might raise an eyebrow, perhaps a little bit more temperamentally than I am doing now, if I was denied hypothermia. 8% is not trivial. So this is a frequentist approach. And the frequentists have ruled the stage for a bit of time, but are being challenged increasingly. And very recently, a group of very prominent scientists from across all disciplines published in Nature a very passionate article signed by over 200 scientists, again, as I say, in all disciplines. And they argued that one should retire statistical significance. And what they said really boiled down to the fact that actually a p less than 0.05 is arbitrary and binary. Moreover, what they pointed out was that actually all you're saying by a p less than 0.05 is that when you repeat the experiment in the same number of patients innumerable times, you have a 5% possibility, just by chance alone, to find the same findings, or at least as extreme or beyond that extreme. It's not 5% chance of benefit or no benefit. It's quite different. And they explained this by this diagram. So the solid line here is the null line. This is the frequentist approach, the one that we most standardly use. And what you see is here are a dotted line, and that dotted line is where just by chance you may get patterns of findings with the point estimates on those line. Now, to rule this one as non-significant because its boundary goes beyond the null line makes sense from a frequentist point of view. However, it doesn't make sense if you think about it, because it's actually encompassing some of that same boundary as in the blue line. So it may be that we're simplistically glossing over this and throwing out some valuable information. And that's really where a different paradigm comes up. And in a non-algebraic form, I'm going to try and walk you through what the good Reverend Thomas Bayes told us in the 18th century. And what he told us, and I'm going to try and translate this because some of the language, unless you're used to it, is a bit intimidating. At least it was to me. So the Bayes theorem propounds that the posterior probability, and all the posterior probability means is that this is looking backwards after you've done the experiment. What is the probability? That is directly proportional to the product of the likelihood, and what the likelihood actually means in everyday terms is what you've found in the experiment, your actual observations, and the prior probabilities. That is probably self-explanatory. It's what you may have thought about as being probable before the experiment was run. Stated in a simpler way that I think is easier to understand, the after probability, the after the experiment probability, or the posterior probability simply combines the observations or the likelihood from a new study with the priors. So how does this unravel? How does this work out? And what are the different paradigms? Before we go back to the Thakpa experiment and try and reinterpret it, what I'm going to try and do is to give you my own distillation. So it's a non-statistical distillation, but it's my distillation of the two paradigms. And hopefully that will help. And I've put them into two slides as a table. They're radically different approaches, scientifically speaking. So the starting point or the direction of the frequentist is to pose a hypothesis. What she then does is to look at the observations, and she asks, are the observations consistent with that hypothesis? In quite contrast, the Bayesian approaches it differently. What he or she does is to look at the observations of the experiment and then asks which hypotheses are these consistent with. So this is, I think you can see the difference when it's posed as starkly as that. The study question is also a little bit different because for the frequentist, it's a single viewpoint. Does the intervention reduce the risk? In sharp contrast, the Bayesian can move in several directions. You all know that what the Cochrane plots do is to amalgamate and pool in a relatively simplistic way. It's been very useful for us. I'm not dumping on Cochrane, don't misunderstand me. But what it does is pool differing results. Some may be in different directions, but it pools them. And then you may do subgroup analyses, but usually Cochrane doesn't correct for the p-value of doing those numerous subgroup evaluations. But having said that, it's really a single pooling, a single viewpoint. The Bayesian can look at it from the viewpoint of what prior probabilities were there. So if you looked at some studies that were skeptical, that showed harm, you can pool those together with your new observations. If you have no prior studies, and there were no prior studies for cooling in this particular group of children, then you're in a neutral or null mode. You're in equipoise. You may have studies that were quite enthusiastic, and so you would pool together with your observations from the study with an optimistic prior. Now, this can get confusing, and it leads to some issues at times, but it has an advantage of being able to pool several sets of data. Moreover, the currency of the frequentist is a p-value as opposed to a probability for a Bayesian. People understand, both clinicians and parents and patients understand probability most likely much, much better than they understand p-values. And moreover, the p-value is constrained because, as I told you, and as you recall, it's simply a distribution of repetitiveness. And so just by chance, you're going to be one side or the other. So if you use up your p spending function, then you're limited to the number of interim analyses. And that is at the bottom here. You're limited by your p spending function. Actually, that doesn't apply for the Bayesian, which theoretically you can do again and again. And I'll show you how this is put into practice in a moment. There's also the issue of how the results are expressed. And for the frequentist, they're in the form of absolute and relative risks, and the benefits are stressed more by the Bayesian. So now, let's go back. So fortunately, the data set of the Thakpa trial was recently reevaluated using a Bayesian approach. And what these authors, some of them including the original authors, but amplified by many statisticians, looked at what the trial could actually tell us. And when they applied an analysis,